Yeah, I'm very happy to talk to you about advancing therapeutic approaches for erythropoietic protoporphyria. The erythropoietic protoporphyria, or short EPP, is a rare disease affecting about 80 patients in Switzerland, which is about 10,000 patients worldwide, meaning an incidence of about 1 in 150,000. So it's quite rare. And it affects heme biosynthesis pathway, which is mainly active in the bone marrow for the production of red blood cells. There, in this disease, ferrochelatase, the last enzyme that integrates iron into protoporphyrin 9, is defect and leads to the accumulation of protoporphyrin 9, which is a phototoxic metabolite. It then spreads in blood, skin, and the liver, which then means for patients when they are exposed to sunlight or other visible UV light, that protoporphyrin 9 is activated and releases energy to form oxygen radicals, meaning that patients experience photosensitivity, often described as a tingling, itching feeling, followed by this phototoxic reaction, which is painful and leads to injuries that are burn-like. Further, this might lead to liver dysfunction due to the accumulation and crystallization of protoporphyrin 9 in the liver. So overall, this leads to patients avoiding sunlight or trying to cover as much of their skin as possible. So next to a physical burden, this also has a psychological burden and reduces overall their quality of life. Up until recently, there was no real treatment, but in 2015, the EMA approved of afamilanotide, which is a peptide drug that is thought to increase pigmentation and thus reduce the burden of light on patients. Likely also its anti-inflammatory and antioxidant characteristics are beneficial. So all efforts rather manage the symptoms than address the actual cause of the disease. Now, what is known about the cause of the disease? This now goes into the splicing of ferrochelatase, meaning the maturation of the ferrochelatase pre-mRNA into mRNA, where exons are spliced together. And in a healthy individual, this is done correctly, meaning that especially exon 3 and exon 4 are joined together. In a patient, the scenario is a bit different. A patient will inherit one loss of function allele in combination with a allele that carries one T2C single nucleotide polymorphism, which favors the use of a cryptic splice site located upstream, and the mature mRNA then includes 63 additional nucleotides with an in-frame stop codon, which will lead to nonsense-mediated decay of the mRNA, and that's where we finally end up having low levels of the ferrochelatase and accumulation of protoporphyrin 9. Now, in the table to the right, the connection of the SNP and splicing is shown. So the C-SNP really shifts this splicing to form an aberrant spliced product. And I want to highlight once more that purely the loss of function allele in combination with the C-SNP leads to development of the disease, as ferrochelatase activity drops to below the threshold of about 35%. A CC genotype or a loss of function in combination with a T-SNP still are considered having a normal phenotype. And this also implies that elevating ferrochelatase levels or activity by just a few percentage might already be beneficial. So to bring a patient from symptomatic to asymptomatic. Now I would like to talk about 3 different approaches that the Hall laboratory has been taken to address the disease. And as the first one, I would like to talk about splice switching oligonucleotides. The oligonucleotide field has already successfully developed splice switching oligonucleotides. And here I want to briefly talk about nusinersen as an example for such an oligonucleotide. In patients, patients have too little levels of survival of motor neuron protein, which is usually expressed from the SMN1 locus. Now, in humans, there is a copy of this gene which differs by a few nucleotides only, but that leads to these certain SNP leads to aberrant splicing, meaning that exon 7 is usually not included in the mature mRNA, and mostly truncated, unstable SMN protein is produced off of this locus. Now, nusinersen is able to correct splicing and promote inclusion of exon 7, and so rescues production of SMN. So not only is this a treatment, but it's also, the splice switching oligonucleotide is also a rather straightforward tool to gain insights into the pathobiology of a disease and opens up the new therapeutic avenues. Similarly, in the Hall laboratory, a splice switching oligonucleotide has been developed that is able to correct splicing of ferrochelatase. This work has been published a few years back, so if you're interested in the details, you can look it up. What I would like to highlight here is that we have a splice assay that is able to semi-quantitatively analyze the amounts of aberrant and correct splicing product in a certain sample by inputting the sample into a PCR reaction, which uses primers annealing on exon 3 and exon 4. This will then lead to PCR products that differ in length, meaning the aberrant splice product will be longer than the correct spliced transcript, and this we can resolve on a gel and such determine the splicing that is happening. So when we look at cells treated with the splice correcting oligonucleotide, well, first of all, here we can see in the first lane of the gel a healthy K562 cell line, and next to it a stably expressing K562 cell line that is showing the splicing defect, as one can see in an intense band for the aberrant transcript. Upon treatment with the splice switching oligonucleotide, we can see a time-dependent correction of splicing as the aberrant band fades out and the correct transcript increases in intensity. Further, in the Hall laboratory, a mouse model of the disease has been created by mating a mouse carrying a loss-of-function mutation in with a mouse carrying a partially humanized ferrochelatase gene, especially in the region of exon 3 and exon 4, as well as intron 3 with the disease SNP. Now, when these are mated, their offspring, some of their offspring will carry the loss of function allele in combination with this humanized allele. Now, in most recent and unpublished work by my talented colleague Phil Becker, he has characterized these mice, and we can see that their amount of correct ferrochelatase transcript in the bone marrow is decreased, as well as protoporphyrin IX levels in whole blood are increased. When these mice are then treated with the splice-switching oligonucleotide conjugated to a transferrin receptor ligand, we can see that correct ferrochelatase transcript is increased, as well as the protoporphyrin IX is slightly lowered. We hope to continue these experiments, and with an extended time frame, we are likely to see an even stronger effect on protoporphyrin IX. So next up, I would like to talk about a small molecule approach for erythropoietic protoporphyria. First of all, in this experiment, an assay to measure protoporphyrin IX induced phototoxicity was set up, as then to use this setup to test various small molecules which might be beneficial in terms of lowering accumulation of protoporphyrin IX and thus increase cell viability upon UV radiation. When a model of EPP is irradiated with UV for about 30 minutes, 24 hours later, their viability is very poor based on protoporphyrin IX induced toxicity. These results are summarized on the right on the graph. So non-treated or edema, so control treated, show poor viability. And as a positive control, we have treatment with succinyl acetone, which is able to increase cell viability, though it is not considered as a viable treatment, as it induces accumulation of another metabolite in the pathway and usually leads to hepatic toxicity, though it serves the purpose to showing how cell viability may be increased in this assay. Then two small molecules, which are inhibitors of glycine transporters, were tested. The rationale being here that reducing the influx of glycine into the heme biosynthesis pathway may reduce the accumulation of protoporphyrin IX downstream. Now, both bitopertin and ORG-25543 are able to, in a concentration-dependent manner, increase cell viability, while ORG-2543 is stronger in its effect. I would want to point out here that bitopertin is currently in phase 2 clinical trials, from which we are looking forward to hear results quite soon. Yeah, and finally, I would like to talk about a gene editing approach that is currently being investigated in the group. Yeah, so CRISPR-based gene editing tools give us the tools to precisely address the genetic cause of erythropoietic protoporphyria. And these approaches have been developed together with the Schwank lab at University of Zurich. And in the first approach, we would aim to mutate the cryptic splice site. And in second approach would aim to revert the disease SNP-C back to a healthy T. You can see the different editors and their aim in the list below, but today I would like to focus on the first approach. Mutating the cryptic splice site, which is shown here in orange. So this approach would be using an adenine base editor, which at its core is a mutated Cas9 enzyme, which is fused to an adenosine deaminase. And this whole complex is guided by the guide RNA to the locus of interest. Once it finds that, the target strand is nicked and the adenine adenosine deaminase is able to deaminate any adenine base in the editing window upstream of the classic PAM sequence. So what does that mean concretely? The adenosine would be deaminated to form inosine, which then would lead to a mismatch, which is likely repaired on the side of the thymidine due to the induced Nic, which would mean that the inosine is then paired with a cytidine, and upon DNA replication or repair, guanosine should be installed, which would mean that editing was successful. So we wanted to test out these editing approaches in a patient-derived cell line, a lymphoblastic cell line. And first of all, we sequenced the cell lines to see whether at the SNP location they really had the phenotype, the genotype. So for the EPP LCL, we have a signal Y, meaning a cytosine or thymidine at the location. Likely the thymidine comes from the loss of function allele and the cytosine from the splicing defect allele. Our control cell lines are homozygous for T and C. We can also look at these cell lines in the mRNA splice assay. And also here, the EPP cell line shows the splicing defect, which is highlighted upon treatment with an inhibitor of nonsense-mediated decay. So it has the least amount of correct spliced mRNA. And finally, we can also look at ferrochelatase levels by Western blot. Here again, we conclude finally that the EPP cell line is a suitable model system as it carries the correct genotype and phenotype. We then went on to nucleofect these cells with editor and guide RNA. And 3 days later, we collected cells to extract gDNA and sequence the region of interest ferrochelatase gene. Upon nucleofection with both the editor and the guide, we can see up to 14% A to G conversion at the cryptic splice site, as well as some bystander edits upstream, which are likely not biologically relevant, but we will need to assess that in following experiments. But it is expected to see these bystander edits as they are located in the editing window upstream of the PAM sequence. So yeah, just to sum up briefly, I hope to have convinced you that we are working on several therapeutic approaches for erythropoietic protoporphyria, which address the cause of the disease, namely the splice switching oligonucleotide, using small molecules to inhibit the accumulation of protoporphyrin 9, as well as the most recent approach using gene editing to address the disease. We are excited to take this approach further into the mouse model of erythropoietic protoporphyria. Here we could be working either with an ex vivo or an in vivo approach, as our tissue of target tissue are hematopoietic stem cells. Finally, keeping in mind that we would like to accelerate the development of novel therapies for this disease. With that, I would like to just thank Phil Becker and Celine Veller, as well as Professor Jonathan Hall, for letting me work on such exciting project, as well as Dr. Yasmin Barman and Dr. Elizabeth Minder at the Trainee Hospital for their expertise on this rare disease, Dr. Tanja Rothgangel and Kim Markart with Professor Gerald Trank at the University of Zurich, as well as Sebastian Siegner with Professor Jacob Korn at ETH Zurich for their expertise in gene editing, both theoretically and applied. And finally, the whole group for creating a fun and wholesome environment to work in. And obviously you for your attention.
Great talk, Ava Maria, with so many great approaches to address EPP. If you guys have any questions for Ava Maria, please click on the Q&A button to type those in, or you can use the raise hand function. For me, I was wondering, for your splice switching oligo, I think you got a 1.5-fold increase in the corrected isoform. Do you think that that would be enough for an effect on symptoms?
Based on what we see, yeah, let me briefly pull up the slide. Based on what we already see, the slight lowering of protoporphyrin 9 in whole blood, I think that it might be sufficient, as kind of slight correction of the splicing might kind of have a bigger effect on activity or correct ferrochelatase and then downstream accumulation.
Okay, great, thanks. We have a question from Elena Feinstein. What is the efficiency of TFR1 ligand-mediated delivery of oligos to erythroid progenitors? And how frequently dosing should be performed given in view of high rate of erythropoiesis?
Sorry, I didn't get the second part of the question, but maybe I start with the first one.
Okay.
So it is known that that the target tissue, so the hematopoietic stem cells, do express transferrin, and yeah, kind of the internalization of the receptor is very promising to have this actually and then delivered to the cells. And yeah, the second part of the question, I did not fully understand that.
I think they want to know how frequent you need to dose because there's a high rate of erythropoiesis.
Yeah, this approach with a splice switching oligonucleotide would likely need more frequent dosing, as yeah, through differentiation of these cells, probably the effect wouldn't be so long lasting. But I cannot really speak to that yet. I guess that would be also, since that would be gained through experimental testing. But obviously the hematopoiesis in mice also works differently than in humans. Finally, I think this all speaks rather to a gene editing approach, as we could relatively easily target hematopoietic stem cells and make sure that these cells then actually provide for, in the long term, yeah, benefit to the patients.
Okay, great, thank you. I don't think there's any more questions, so maybe we can move on to Josh.
Hi. Yeah. So yeah, hi. My name is Jathavan Asohan, as I've introduced earlier in this presentation already. And today I'll be giving my talk on self-assembled nanoparticles for the delivery of functional nucleic acids. So, of all communities, I don't need to attest to the value of nucleic acids as therapeutics. Whether you're starting with antisense oligonucleotides in the 90s, or moving on to siRNA, and most recently mRNA, nucleic acids have shown to be invaluable in the treatment of disease. addressing diseases that previously could not be, or addressing them in novel ways. And there's been a lot of improvements to NATs along this pathway. Whether you're looking at backbone modifications such as phosphorothioates or thiophosphoramidates, or base modifications to increase hybridization, such as 2'-methoxy, locked nucleic acids, or 2'-fluororabinose. We've come a long way from using basic phosphorodiester DNA as a therapeutic. However, despite the fact that we've come so far, we haven't cleared all the hurdles of nucleic acid therapeutic to nucleic acid therapeutic delivery. We still suffer from issues such as biodistribution and cellular penetration. In order to address these challenges, we propose the use of nanocarriers. So nanocarriers are nanoscale systems that allow for the hiding of the nucleic acid molecule itself and presentation of the molecule as something else once entered into the body. And this presentation can differ very greatly, either by changing size, changing shape, having different materials as the other core, or encapsulating the actual therapeutic, or by changing the surface properties of the therapeutic itself through pegylation, changing of the surface charge, or introducing targeting ligands to get better delivery towards tract tissues. But however, using nucleic acids, we're able to access a very interesting library of interactions. So a lot of times you can look at DNAs like Legos. A lot like Legos, we've got these very specific interactions between complementary base pairs, forming tight, specific interactions. Much like when you take two Legos together and you push them together, you get a very specific interaction that's very tight and very hard to break apart. And using Legos, we're already able to create such interesting designs in the macro scale. So whether you're making a small house or this incredibly complicated full-on building. In similar ways to this, we're able to use DNA as a material in order to make either simple or complicated designs. Whether we're looking at assembly of individual tiles into these large macroscopic but rather simple shapes, or we're able to use DNA origami to specifically fold a large viral RNA into a specific shape. Using DNA as a material for creating new structures has been very well studied. However, with Legos, it's a lot more interesting when you introduce other modalities. So whether you use rubber bands to power a Lego car, or whether you use glue to hold together structures that are going to be exposed to a lot of people, adding more functionality externally outside of Legos allows you to increase the actual usability of the Lego itself. And the same applies to oligonucleotides. As pioneered by Dr. Tom Edwardson in our lab, we've been able to conjugate various non-canonical DNA-like structures containing diols onto nucleic acid strands with a very specific function with very specific structures. And a brief overview of how we do this itself is using solid phase DNA synthesis. So starting with a controlled glass bore bead, we're able to sequentially add monomers one and then two at a time until we're able to create a full nucleic acid strand. Then taking the strand, we're able to add our hydrophobic blocks or other monomers, adding sequentially until we're able to create our final polymer. And this synthesis creates a very monodispersed and sequence-controlled product. And this specific structure that we're going to show here contains two different moieties, 12 hexaethylene repeat units conjugated to a nucleic acid strand. In our cases, this nucleic acid strand is an antisense oligonucleotide for luciferase that I will be going into later. But taking the strand and putting it into a magnesium-containing buffer, we're able to self-assemble these strands into discrete structures that we deemed spherical nucleic acids, specifically self-assembled spherical nucleic acids. And this is work that was done by Dr. Tom Edwardson and Dr. Nanny Busmail, and was built upon by both Dr. Nanny Busmail and Dr. Asohan in order to showcase the usability of these spherical nucleic acids in cellular systems, whether we're looking at in vitro with triggered release of a therapeutic, or we're looking at the preferential distribution of these spherical nucleic acids. We have a lot of promise with these structures. However, one question arose that was, looking at small modifications of the structure. Introducing such a different moiety in of itself, we created one structure, but in theory, can we change the structure further? And in this structure, we have entirely phosphate linkages, but we proposed the introduction of phosphorothioates to the core itself. This was done in order to increase the hydrophobicity of the core itself. And since the self-assembly of the structure is dependent on the hydrophobicity of the core, we proposed that this would create a more stable structure that would then be able to deliver better. And as such, we created these two different nucleic acid, these two nucleic acid strands, one containing a phosphodiesterase core and one containing a phosphorothioate core, both once again containing the luciferase antisense oligonucleotide as the DNA portion. And we can see really clearly via dynamic light scattering that we have similar sized SNAs formed in both cases, all ranging between 14 and 17 nanometers in radius. Furthermore, looking at atomic force microscopy, we're able to see a very mono-dispersed product. A lot of individual spherical structures indicating that we have assembly of spherical nucleic acids once more. Next, we wanted to look at the actual stability of these structures when it comes to changing conditions. So when we assemble these structures, as I mentioned before, they're done in magnesium conditions, in high magnesium, in very controlled setting. However, that's not what we're going to experience when we have them injected systemically. We're going to experience a decrease in pH and a decrease in magnesium. And as such, we tested the stability of the cores of these structures using a fluorescent dye that intercalates into the core of the structure. And then by looking at fluorescence of this dye, we're able to study the stability. And interesting, between the two structures, we see quite a difference, with the phosphorothioate core structure showing less stability overall, especially in low pH, low magnesium conditions, which is great. Our primary structure dictates stability of the overall supramolecular assembly. Next concern we wanted to address was the perils of serum proteins. So serum is not, once again, just a simple buffer like we use to assemble our structures. Proteins in circulation bind to nanostructures, opsonizing onto the exterior, allowing for different presentation of the actual structure itself. Furthermore, binding of serum proteins, and specifically albumin, has been previously studied in our lab by Dr. Erythropoietic, and has been shown to decrease uptake and have many other effects that may or may not be advantageous to us as we move forward. Ultimately, this means our self-assembled SNAs can undergo two different fates. Owing to the fact that they are held together non-covalently, they can either remain intact with a protein corona formed by albumin or other proteins, or we can have disassembly into the single strand itself, with the single strand's hydrophobic interior binding specifically to the interior core of albumin, resulting in this disassembled structure. And when we do agarose gel electrophoresis, we see the presence of what we propose are both constructs. We see a higher mobility band, which we think is indicative of the disassembled structure, and a lower mobility band that is the spherical nucleic acid itself with a protein and albumin core. So knowing that we have both intact and disassembled states presence, we want to look at the effect, and the effect itself also differs depending on the oligomer used. However, in both cases, in maintenance of the spherical nucleic acid promotes entry into cells. Using a fluorescence, using flow cytometry, we were able to show that by forcibly disassembling the structure itself, by pre-binding to albumin prior to incubation with cells, decreases uptake compared to introduction of the entire spherical nucleic acid to serum. And this is true of both structures. Ultimately showing us that the cis-SNA character is key to actually internalization, and the majority of the internalization comes from this spherical modality. Next, we looked at silencing. So moving from using our phosphorothioate DNA to a phantom-modified strand, we're able to show unaided gymnotic silencing within 2D cell culture. This was done, once again, using our antisense oligonucleotide for luciferase and measuring the decrease in luminescence associated. And we see a significant difference in the silencing ability between the two structures. So despite all their similarities in terms of cellular internalization, in terms of structural assembly, we see that minute changes in core affect silencing, with phosphorothioate core itself specifically increasing silencing the most. So we've come a long way. We've shown that our spherical nucleic acids can silence unaided, and we've shown that we can have preferential silencing based on the structure, and that the spherical nature of our structures helps in internalization. However, can we stabilize this spherical shape further? And by doing so, can we increase silencing further as well? In order to do this, we introduce other supramolecular modalities, specifically G quadruplex. So for those who don't know what a G quadruplex is caused by the interaction of four guanines around a positively charged cation, and is very well recognized as a stable structure in biological conditions. So taking this modality into our spherical nucleic acids, we created what we call the DNA G4PS core structure, introducing 4Gs between the DNA and the hydrophobic core portion in order to create a self-assembled structure that is held together more rigidly. This will be done through the use of the guanines themselves, helping bind very tightly along the core of the structure, owing to their pre-assembled nature by the SNA. Once again, looking at characterization, we see similar sizes to previously, a little larger, more along the lines of 17 nanometers, as opposed to the 15 previously mentioned. And very interestingly, looking at circulated chromosome, we see the emergence of a peak between 250 and 300 nanometers, indicative of the formation of a parallel G quadruplex, significantly more than the structures lacking this G4 modality. And furthermore, looking at the effect in cell culture conditions, we see that the presence of this upper band that we associated with the intact SNA bound with a protein corona increases. We're able to promote more of the actual intact structure remaining in solution than when we do not have this G quadruplex. And furthermore, looking at internalization, we see positive effects by the introduction of the G quadruplex. Over the course of 72 hours, we see significantly more internalization than we do of the for the non-G quadruplex containing structure. So ultimately, we're able to show here that maintaining SNA character does increase uptake. However, our main question is, does this translate to silencing ability? And interestingly, it doesn't. So while we have increased internalization of the G quadruplex containing SNA, this increased internalization does not come with a subsequent increase in silencing ability. In order to probe this, we looked at the rate of release of our therapeutic antisense oligonucleotide from the actual cores themselves. And here's where we see an interesting difference. The G quadruplex containing SNA shows less overall release and at longer time points, slower release than the structure lacking the G quadruplex. So while we're having more internalization, we're having less release of our functional nucleic acid, that which we've shown previously is important for actual silencing ability. So this, after wrapping up this project, I wanted to once again go back to the concept of Legos. So I want to real quick bring up the concept of illegal builds. So this is what we call a legal build in Legos. It's when you have the actual Lego bricks parallel to each other, and then you're able to click them together. And you form this very tightly formed, very rigid structure. However, you can also have illegal builds when you don't use the studs on the Lego bricks as intended. Instead, you introduce orthogonal interactions using either the side or other portions of the Lego bricks to create structures that are still stable, but not as intended. And we're actually able to use the same thing with nucleic acids. So we all know about the interaction between adenine and thymine through the use of the Franklin Watson-Crick base pairs using the canonical base pairing system. However, as discovered and shown by Dr. Nicole Avakian in our lab, in addition to the Franklin Watson-Crick base, adenine also has a second phase, a Hoogsteen phase. And through this Hoogsteen phase, it can bind to other thymine-like molecules, creating novel structures that are not used in biology presently. And using this Hoogsteen phase, we're able to interact with, interestingly, cyanuric acid. So cyanuric acid is a non-toxic molecule that's commonly used in swimming pools. And it has an interesting property in that it is entirely symmetric and contains three different thymine-like phases, 1, 2, and 3, all of which that can interact with adenine and specifically polyadenine DNA in order to form these very interesting and complex cyanuric acid fibers, holding together three different strands of polyadenine with free cyanuric acid surrounding them and allowing for them to come together in this beautiful fiber motif. And we're able to look at these fibers themselves through the use of atomic force microscopy, looking at the structures forming these long network of web-like fibers all along the actual AFM puck itself. However, what Dr. Christophe Legendre and I were able to look at as we moved forward was the similarity of these fibers to fibers that are used for hydrogels. So hydrogel networks are proposed of long fibrous protein or polymer strands held together by cross-link points. And by introducing these cross-link points into our cyanuric acid polyadenine fibers, we're able to create a similar kind of motif using a fiber with a cross-link region. And using these, we're able to create hydrogels. And these hydrogels have blown us out of the water with how effective they've been. In terms of their actual mechanical properties, they've far exceeded other hydrogels that are currently on the other DNA hydrogels that are currently being used, which max out at roughly a G prime of 1,000. We show roughly 100-fold increase in the actual rigidity of our hydrogels. Now, most importantly for OTS, these hydrogels, being entirely nucleic acid, have the ability to encapsulate and deliver therapeutics. And so using a antisense oligonucleotide for luciferase, as we've done before, we looked at luciferase silencing. And interestingly and very, very coolly, we're able to show an increase in silencing ability when you have an incorporation of this modified ASO into the hydrogel, whether we have it just free in solution or it is integrated into the actual hydrogel network itself, we see an increase in silencing ability across the board. And so ultimately, we're able to use the hydrogel for ASO delivery and to increase silencing using this novel structure. So in conclusion, I hope I've shown to you that we can use non-canonical DNA interactions to allow for formation of different nucleic acid structures. And we can harness these structures for delivery and silencing in modular ways by changing how they present supramolecularly. And that brings me to the end of my talk. Thank you everyone for coming and for attending and for listening to all of us. And yeah, I'm open to any questions.
Ooh, Crystal, you might be muted.
Thank you so much. Great job, Jath. I mean, really cool to hear how you kind of took a negative result and built on it to pivot into a more effective platform. I have a broad question. For your spherical nucleic acids, you have so many different properties to play with, like you said, size, material, shape. How do you decide what to manipulate?
It was very much a limitation of our synthesis itself. So at the time we started, phosphorothioates were just beginning to be used in my group, and they're very cheap and very commonly used in actual applications themselves, like in clinical applications. So it very much seemed like a logical progression. And additionally, the switching of the oxygen for the sulfur, it has a significant effect on hydrophobicity. So you can measure this even through high performance lipid chromatography, like just these 12 modifications of a single atom changes the hydrophobicity significantly. So it started there, but we've expanded a lot since, introducing more monomer sequences, going from just using DNA to siRNA, and including adding other molecules to the actual surface of the nucleic acid to change how delivery occurs.
Very cool. Okay, thanks. We've got a couple of questions in the chat. And if anybody else has any questions for Jeff, feel free to raise your hand or go to the Q&A button. Mark Antoine Turcotte says, did you look at the effect of adding the lipophilic chain and 4 guanines on the binding TM of the oligonucleotide by UV melting or other techniques?
Yes, so we've looked at the melting temperature of the SNA itself. With the G quadruplex containing structure, we have done some studies and we've not seen a significant difference in the actual melting temperature itself, which is kind of surprising to be like, there's a couple of competing factors that we were debating when we were looking at the actual potential effect of TM, whether the increased bases would sterically block it or whether the rigidity would decrease the melting temperature. But at present, we haven't observed any real difference between the G quadruplex containing strand and the non-G strand.
Okay, great. Pedro Moreno wants to know, he says, thank you for the interesting presentation. Which mechanism do you think is taking place for the oligo uptake mediated by the hydrogel system? In principle, it is still a genotic delivery. Is it cell behavior and presence of the gel?
Yes, that's a great question, and that's something we're actually actively looking at. So, what's interesting here is this actual structure itself is very pH dependent. And over the course of the timescale of the experiment, as we tested it, we see the hydrogel itself flow, like disperse, but the effect of the hydrogel itself is very prominent in comparison to not having the hydrogel. So what we propose is actually there is no difference in internalization, but upon internalization and acidification of the endosome itself, we're able to kind of disrupt the endosome and get better delivery into the cytosol. And that's something we're actively looking into right now.
Okay, great. If there's no more questions, I want to thank Jath and Ava Maria for some really cool presentations showcasing what they've been working on, and really nice to hear what they're interested in contributing to the OTS community. I know that me and Jillian have had a really fun time being immersed in this society and getting to see the inside workings and also try and give back by contributing what we can. And I'm really excited to spend this next year together. We have more webinars coming up, so feel free to check out the schedule online on the OTS website or on the OTS LinkedIn page. And these webinars are all recorded, so feel free to come back and watch these online or email Ava Maria or Jathavan for any questions you guys still have.
Thanks, everybody.